Okay, students, so this is industrial biochemistry, and the course code is BCHM351, and this is lecture 2, and the topic is production of penicillin via fermentation, part 2. And I am Dr. Musaddiq Ibrahim, Assistant Professor, Department of Chemistry, Kohart University of Science and Technology, and you can access all these lectures through the Cust Media Server portal, whose website is www.ms.cohort.edu.pk. Now, moving towards the topics which will be discussed in this lecture. So, we will be discussing the methods for antibiotic production, that how the antibiotics are produced and what are the methods which are used for the production of antibiotic. Then is the penicillin production by the process of fermentation. Along with it, we will be discussing downstream processing. So it is a part of the fermentation process in which the final product is being refined, purified, and extracted out. So we will be discussing the downstream processing in detail. Also is the operation in the fermentation tank, that how the microorganisms are being put in the fermentation tank, and then they are provided with specific food nutrients through which they can grow, and as a result their metabolites also grow, and indirectly the penicillin, which is a secondary metabolite, that is also being produced by the microorganism, and then later on it is being purified, extracted out from the fermentation tank. Also, we'll be discussing the varieties of penicillins. So there are different types of penicillins which are in use nowadays, so we will be discussing them as well. Now, moving towards the antibiotic production methods. So, like all antibiotics, penicillin is a secondary metabolite, and it is only produced in the stationary phase of the growth curve of the microorganism. So an important thing here to note is that all the antibiotics, these are the secondary metabolites of the microorganisms. Now we will also be discussing that what sort of fermenter does it require. So in case of penicillin production, it requires a batch fermenter, and along with the batch fermenter, a fed batch process is used so that the stationary phase is being prolonged, and as a result of a prolonged stationary phase, the production of the secondary metabolite in the form of penicillin is also enhanced. Now moving towards the downstream processing. So what is downstream processing? So products in a fermenter are impure and dilute, so they need to be purified by downstream processing. Now what is happening in downstream processing is it involves the filtration to separate the microbial cells from the liquid medium, and along with it there are a couple of chemical steps which are for the purification purposes as well as for the concentration of the product. Now downstream processing can account for 50% of the cost of a process. Now, the downstream processing. So, downstream processing is a relatively easy task since penicillin is secreted into the medium to kill other cells, so there is no need to break open the fungal cells. So this is an important advantage of the production process of the penicillin, that when the penicillin is produced as a secondary metabolite, so it is being secreted outside the bacterial cell into the fermentation tank. So in that case, the penicillin is already present outside the fungal or bacterial cells. So um, the cells, they don't need to be damaged or uh, destroyed or to be broken. So along with it, the importance of downstream processing comes in action, that as a secondary metabolite in the form of penicillin, and the penicillin will be used as a antibiotic, as a drug for treating patients. So therefore it needs to be very pure, as it is used as a therapeutic drug. So what is carried out is that the fermentation tank, within the fermentation tank, all the penicillin along with the liquid medium is dissolved and then precipitated as potassium salt. Now it is precipitated as potassium salt. The purpose is to separate it from the other substances in the medium. Now the graphical time course of penicillin production. So in this graph, you will understand that what is happening within the fermentation tank when it is subjected to penicillin production. So on x-axis we have the time duration in days, and on y-axis we have the amounts of nutrients or antibiotics or cells either increasing or decreasing. So first of all, we will focus on the cells, the fungal cells which are producing the penicillin as a secondary metabolite. So here you can see that in the start there is a lag phase, and then after about 1 day within the fermentation tank, the cells, they start to produce, to uh, multiply or increase exponentially, and ultimately they reach to a point when after 3 days within the fermentation tank, during the process of fermentation, the stationary phase is started from this point. Now the important thing is to focus, is to observe that when the stationary phase has started of the fungal cells, so at this point, slowly and gradually, the secondary metabolite in the form of penicillin, it starts to be produced, and ultimately it goes to the peak of its production along with the stationary phase of the fungal cells. So you can see that uh, during the process of the stationary phase, the penicillin is being produced, and along with it, here we are talking about lactose, which is a carbohydrate source for the fungal cells to grow in number. So in the start, the lactose concentration starts to decrease. So at the beginning, the maximum amount of lactose is provided within the fermentation tank, And as the fungal cells are growing, so does the lactose concentration is decreasing. And with the passage of time, after about 4 or 5 days, the lactose concentration is totally depleted within the fermentation tank. So it means that the lactose is being utilized by the fungal cells so that they can grow, and as a result, along with the growth of the cells, when they reach to the stationary phase, the penicillin production, it also starts. And you can see that after about 7 days, the maximum penicillin production is being achieved within the fermentation tank. Now moving towards the flow sheet diagram of penicillin production by the process of fermentation along with the downstream processing. So this is the flow sheet diagram for the penicillin production, and here we have the batch fermenter. So first of all, the starter culture, which is composed of Penicillium chrysogenum species, that is being added to the fermentation tank. Along with it, the nutrition or the medium is also added at the start of the fermentation process, and the medium or nutrients, they include corn steep liquor, that is basically sugars and composed of lactose. So these are the sources of carbohydrates. Along with it, yeast extract is also added as a medium into the fermentation tank, which is a source of nitrogen, and you can say that it is a source of proteins. The pH buffers are also added so as to maintain the pH of the fermentation tank within the batch process. And some minerals are also added at the start of the fermentation process. Now, as a result of starter culture along with the medium, the nutrition provided, the culture becomes 10 times in 6 days. So it means that with the passage of time, as we discussed in the previous slide, that in the timeline growth of the microbial culture, so we have seen that as the cells, the fungal cells, they start to grow, so does the lactose concentration decreases, and ultimately when the fungal cells they reach to a stationary phase, then the production of the penicillin starts. So you can see that the culture becomes 10 times in 6 days. Now what is carried out is that remove 30% culture from the fermentation tank and then add 30% fresh medium. So the medium is again added after 6 days uh, after the removal of 30% culture. So the purpose here is to provide more medium to the culture so that within the stationary phase, as we know that after 3 days the fungal cells go into stationary phase, and during the stationary phase the secondary metabolite in the form of penicillin is getting produced. So when you are adding 30% fresh medium, so it is being provided to the fungal cells who are still in the process of growth, and they also reach to the maximum uh, growth level and enter the stationary phase, and as a result more and more penicillin production is started. Now there is a rotating filter after this process, and the rotating filter is separating the fungal cells as well as the filtrate from each other from the culture media. Now the fungal cells, as they are of no use because the fungal cells have secreted the secondary metabolite in the form of penicillin, and that is in the filtrate. So the fungal cells are used as animal feed. Now the filtrate, it contains the important ingredient, which is the penicillin, and here the downstream processing steps are being followed, which is, first of all, the filtrate is dissolved in butyl acetate. Then afterwards the potassium ions are added to precipitate the salts of penicillin. Now then is the wash, filter and dry step, after which you can get 99.5% pure penicillin. After you are getting the pure penicillin, there are further steps which include chemical and enzymatic modification to make new antibiotics or modified form of the penicillin, like ampicillin, amoxicillin, and other derivatives of penicillin. Now moving towards the operation in the fermenter. So this is the general structure of the fermenter and its different parts. So first of all is the gases out. So there is an outlet which is responsible for the gases which are produced during the process of fermentation to let them out through this outlet. Then there is a sterile air inlet from where the sterile air is introduced inside the fermenter. As we know that the process of fermentation in the process of penicillin production is an aerobic process, so that's why the sterile air is provided into the fermentation tank. Along with it, the nutrients are also added, so there is an inlet through which the nutrients are added into the fermentation tank uh, during the start of the fermentation process. Then you have a temperature monitor which is situated here inside the fermentation tank, and the purpose of monitoring of the temperature is that there is a specific temperature at which the fermentation process takes place, and maximum yield can be obtained. So if the temperature either decreases or increases from that optimum temperature, so then there is a availability of cooling water in and cooling water out around the fermentation tank, so that the fermentation tank will not get heated up. There is a provision of stirrer along with the stirrer paddle, uh, which you can see is here, and the purpose of the stirrer and the stirrer paddles is to uniformly distribute the nutrients and the media within the culture, so that all the fungal cells can have access to the media, and so they can grow as maximum as they can, and then they can enter into the uh, stationary phase. There is a ring of air outlets. So we have discussed this, that for the uh, entry of sterile air into the fermentation tank, there are rings which are present, and as a result of those rings, the air bubbles, they are introduced into the fermentation tank. There is also the provision of pH monitoring. So the fermentation process takes place at an optimum pH, and that pH is being monitored through a device which is inserted into the fermentation tank. And if the pH monitor shows that the pH is not of optimum value, so then there is an inlet for adding acid or base into the fermentation tank. So if the pH has decreased, then the base will be added in order to get the pH of the whole culture to a optimum pH level. And if the pH of the medium within the fermentation tank has decreased, then the if it is increased, then the acid is added so that the pH in the fermentation tank may be um, optimized to the normal values. And the last one is the antibiotic out. So there is an outlet from where the antibiotic can be obtained from the fermentation tank after the process of fermentation has completed and the time duration has been completed, that the maximum amount of the secondary metabolites in the form of penicillin has been produced. So then the antibiotic is taken out. Now moving towards the varieties of penicillins. So the resulting penicillin, which is produced by the process of fermentation, is called penicillin G, and it can be chemically and enzymatically modified to make a variety of penicillins with slightly different properties. So it means that after the process of fermentation, the end product is penicillin G, but then it is chemically and or enzymatically modified to form other varieties of penicillin. So these semi-synthetic penicillins, they include penicillin V, penicillin O, ampicillin, and amoxicillin. Now there are some important questions which you should be able to answer after listening to the part 1 of this lecture and then this part of the lecture. So these questions include that what is the carbon source during penicillin production? So these are trivial questions, and if you are able to answer these questions, so it means that you have studied and listened to the lecture properly. Another question is the nitrogen source during penicillin production. So as I discussed it in the fermentation process of penicillin production, so you should be able to answer that as well. What is the energy source during penicillin production? So that is also available. The answer is available in the lecture. Now, the penicillin fermentation process is aerobic or anaerobic? That is another question. And what is the optimum temperature for penicillin fermentation? So you need to look for the answer of this question. Now some other important questions include that penicillin is a primary or secondary metabolite, and what is the volume of fermenter used in penicillin production? So you need to look up for the answer of this question. And why isn't a larger fermenter used in penicillin production? That is also an important question. And when is penicillin produced during fermentation? So as penicillin is a secondary metabolite, so you know that when the secondary metabolite they start to produce. And how long penicillin can be produced in a single batch? So you should be able to answer that as well. Now some other questions are that What was the first fungus which was known to produce penicillin? So here we are talking about the species of penicillin which was used in the Second World War, and what species produces about 60 milligram per decimeter cube of penicillin. So you need to look up for this answer. How did scientists improve the yield of the penicillin? So there is a specific process or processing. So you should know the name of that process, and that is the process by which the penicillin yield is being improved. What is the substrate used in penicillin production? So here we are talking about the parts of the nutrition or media. And why is batch culture used in penicillin production? So as we know that we have batch culture and continuous culture. So why we are using batch culture? So you need to look up for the answer of this question. Now some other important questions which will clear your concept about the process of fermentation and the production of penicillin through fermentation. So first of all is that during downstream processing, what are the three processes which are carried out in downstream processing? So this I have discussed in the general layout of the fermenter. Now why can't penicillin be taken orally? You need to look up for the answer of this question, and name the form of penicillin which can be taken orally. That is a trivial question, so you should be able to answer it. And how does penicillin kills bacteria? So uh, you need to look up for the mechanism of action of penicillin. And why are gram-negative bacteria not killed by penicillin? So that is also a conceptual question, and you should be able to answer it by looking it up online or through consulting relevant course books. Now the references of this lecture, they include the Austin Principles of Shreve's Chemical Process Industries, and the Handbook of Industrial Chemistry and Biotechnology by Kent and Riegel. Also the Introduction to Cane Sugar Technology by G.H. Jenkins, and Comprehensive Biotechnology by Young and Cooney, and Peter F. and Stanberry, Alan Whitaker, along with Stephen Hall. These are the authors of Principles of Fermentation Technology. So these books are references, and they should be read for further understanding of the process of fermentation technology and the production of penicillin. Thank you.